Hello, everyone. Welcome to the third annual TMT Symposium. My name is Ryan Baumgarten, and I'll be hosting the live webinar. So with me, I have all of our presenters that presented during today's session. So they'll be here available to answer any questions you have specifically regarding their talk, but also some general questions about the future of TMT technology, quantitative proteomics, as well as other advancements in the field. So with that, please use the Q&A to post your questions, and I'll do my best to direct those to the specific speakers. So please use the chat function, and we'll answer them as they come in. I also have some other questions that have already been posted, so I can go through the panel to see what they have. So again, so with me I have Dustin Frost, I have Fernando, and from the Fernando and Antonio, we have Jeff Savas, Stephen Gigi. We have Alexi sitting in for Daniel, as well as Tomas from Pelago Biosciences. So again, if you have any specific questions about their talks, please feel free to use the chat, and we'll answer them as they come in. So maybe just to kick this off, I'll go around and kind of ask some questions that I had for the speakers regarding their talks. So maybe I'll just go in the order of the presentations for today. I have one here for Dustin. I know you presented on a new TMT format where the product is stabilized in a liquid format. Mm-hmm. Are there any comments that you can provide about using that format with maybe like automated systems or just in general, how is it different using the DMSO-based solutions versus traditional acetonitrile?
Right. So, hi, everybody. I'm Dustin. So in developing this, we found that a lot of our users already reformatted our TMT reagents into plates, and they would use acetonitrile, and they might find that when they were doing their automated workflow over time, they could see that that might evaporate. And then also the liquid handling equipment, the pipetters, didn't always accurately aspirate and dispense acetonitrile. So we know that DMSO, with its high viscosity and low volatility, was kind of ideal for liquid handling and would maintain its volume, would evaporate. And we were able to show that the performance of DMSO and acetonitrile was equivalent in our hands. So we think that people will find great success with DMSO versus acetonitrile.
All right. Well, thanks for that. Mm-hmm. I have one here for Jeff. Yeah, really great talk about protein turnover and different protein half-life, I would say, in different regions and cells. So specifically, I know you did contrast kind of the protein half-life in certain brain tissues as well as maybe compared to liver. Have you been able to look at the protein turnover in different subregions of the brain or different specific neurons with your technology?
Hi, Ryan. Good question. Yeah, we have. We've compared a variety of various brain regions, and we don't see really any stark differences between the brain regions. And what we do see more differences in the cell type. So in long-lived cells like neurons, we see proteins have longer half-lives, and the existence of these extremely long-lived proteins that we've reported on. We've really seen our strongest signals in specific compartments. So like the core of the nuclear pore, or chromatin, or mitochondria cristae.
Okay, thanks. I have another follow-up question on yours too, because I know a lot of your mouse studies were using N15 instead of a specific labeled amino acid, like say like a silac amino acid. So is the isotope envelope can be quite complex. So can you maybe comment on some of the software tools that you use to help analyze these types of labeling studies as well as in combination with TMT?
Yeah, so for the N15 metabolic labeling, so it's spirulina algae that's enriched in N15, and we're essentially providing amino acid precursors. And to analyze that data, we rely on software that was developed in John Yates's laboratory or Mike McCoss's laboratory. This was like years ago. And we were limited to identifying the fully nitrogen 15 containing or fully N14 containing MSMS spectra, but at the MS1 level, we have some ways to look at these chimeric species. In terms of the N15 versus the heavy amino acids, yeah, we would love to do those experiments with the heavy amino acids, but the lysine +6 is twice as expensive. The +8 is like 1.5 times, and the heavy lysine ARG is like off the chart expensive. So it's just a practical reason that we use the nitrogen 15 spirulina. Yeah,
no, it makes sense, especially if you have to feed them over multiple generations, as I know you've mentioned before. All right, so let's move to the next speaker. I had another question come in. For the guys at Pelago, either one, so Alexi or Tomas, I think you really highlighted the need to look at multiple cell lines, specifically when you're looking for profiling specific targets, because maybe the targets may be present in some cell lines, but not others. Maybe can you also comment on, is there, in your data sets, do you guys actually normalize for protein abundance? What I mean by that is, let's say you're going after a particular kinase or an HDAC, and in one cell line, it may be at a different abundance level than another cell line. So the target's present, but it's present at a different concentration. How do you guys correct? Do you correct for that, or is this something that you just you see in your final data sets?
I can probably take that. So what we're looking at our data is changes, treatment versus DMS or control. In that respect, the abundance is not playing a crucial role, but depending on the cell line, the protein can be in different conformation in different compartment, and thus it might have different accessibility level for the compound. So that will play the crucial role, the readiness of the protein to interact with the compound, depending on in which system it is.
Yeah, it makes sense, right? So it's not just abundance, it may also be accessibility, right, to the compound. Makes sense. All right, no, thanks, Alexi. All right, let's get everybody involved here. So Fernando or Roberto, thank you so much for your in-depth discussion of all the different experiments of putting the new Accelerome instrument through its paces. What surprised you about the new system in terms of abilities? Can you maybe talk about that? And also, I know you also mentioned, some of your studies that there were some things that you had to maybe troubleshoot or get up to speed with, but maybe you could just get a comment, on your use case. It looked like the data quality in your lab was really great for the samples that were produced, but maybe you can just talk about the ease of use of the system or other things that may have surprised you about using it.
I'm sorry, could you repeat the question? Because, the sound here is not very good.
Sure. I was wondering if you could maybe comment about the Accelerome system, about some of its features or benefits that surprised you, that you were not expecting, or anything about its performance based on the slides that you presented today.
I think that the Accelerome platforms, the output was very positive for us, because it worked. It performed the digestion and the labeling procedure with very high efficiency, and the output with the samples was very nice. We also were very surprised with the results obtained with the micro spectrophotometer, which is incorporated in the Accelerome platform, because and the results obtained there was very accurate. And I think that the only negative point that we have found is this contaminant that we have seen in some samples, these huge peaks that we detected in one of the case studies. And we believe that maybe this is because the presence of a contaminant in one of the buffers that is used in the kits. But in general, our summary is very positive with the instrument. We believe that is a very useful tool for a proteomics lab.
Yeah. No, thanks for that. Yeah, I noticed that. I saw that in your talk as well about some of this TMT labeled, species at the beginning of the gradient, and we actually see this pretty commonly depending on the sample type. So again, you don't see it in every sample, and sometimes there's maybe a small dipeptide or other small molecule that's being labeled by the TMT tags, and it may not be removed efficiently during the cleanup. But I think, overall, I think most of the chromatograms that you showed, it wasn't present, but a few samples it did show up. And we've also seen that in the past with certain sample types. So it's interesting how to efficiently remove excess TMT reagents. And so also I have a couple questions that have come in for Steve. So I have one myself, and I know you presented on GoDig, right? So this target different. Wondering how
I think my computer is choosing at the moment to have a Wi-Fi problem. Let me try to fix it.
Okay, we can hear you.
Oh, you can hear me good. So
Yes.
I did not hear the question.
Okay. Question is, how deep can you go with GoDig and that?
How deep can you go with GoDig? I'm still fighting with my computer here, but I think anything you've seen in the past. So the way we think about it is that if I have collected a data set that has 10,000 proteins in it from a fraction of the data set, any of those 10,000 proteins would be available to you. And we haven't tried to say for the bottom 1% of the proteins, can we still see those or work on those?
Okay. No, thanks. That answers my question. The other question that came in from one of the attendees was, how many proteins can be deposited into the target protein assay builder? And can you still perform GoDig if proteins are not identified by TMT?
Right. So for this question, I get a lot of feedback on my computer, but for this question, I mean, the answer is that we have a mouse, a human, and a yeast library right now. And the human has probably 10,000 or more proteins that are in there. So any of those you could select. But if you cared about a protein that wasn't in there, we're currently evaluating the use of, say, predicted spectrum and predicted order elution. And it seems to be working really well. For example, we just remove a protein from the library and then predict everything about that protein. And 90-something% of the time, we can still quantify it. So there may be a time in the near future when you won't need a library at all. You'll just do everything through.
Oh yeah, that's kind of lost you there, but I think you addressed the question. I think that's great of using, predicted retention times and things like that to find your targets. Let's see. We have another question that came in about benchmarking of the accelerome versus manual sample preparation. So I don't know if Fernando or Antonio want to chime in on that. I know we have done it in our lab. We developed the EasyPep manual sample preparation kits, and there's an application note that does benchmark it against accelerome. But maybe Fernando or Antonio, you can comment on benchmarking the performance of the accelerome versus a manual sample prep, and if in terms of protein or peptide ID numbers, how you felt it performed.
So your question is that we have tried the EasyPrep with the accelerome.
Yes. Have you benchmarked? Yeah. What do you think about the protein ID numbers compared to the manual process?
I mean, they were similar. For example, in our second case study, in which we analyzed 4 different strains from Listeria monocytogenes and Listeria valentina, we already performed first-hand experiments and manual experiments, and the numbers were very similar in both cases. You have to consider that the size of the genome of Listeria is really very small. It's about 2,400 proteins. So maybe someone in the audience has thought that the numbers presented in our presentation were low, but that's because the genome size is small in Listeria. Mm-hmm. So what we have seen is that in this particular case, the numbers between the Acceleron platform and by manual analysis or sample processing were very similar.
All right. Thanks, Alberto. So I guess now these questions are kind of opened up to everyone, and we can go around if people want to comment. But the first one I have is there's a lot of benefits, I would say, to using mass tags and TMT in particular to multiplex samples. So for relative protein quantitation. So which benefit, it could be throughput, signal boosting, fewer missing values. Which benefit do you feel has had the largest impact on your work? So maybe Jeff, do you want to kick us off?
Sure. For me, I think there's 2 huge benefits. Everyone knows this, but number one is measuring the same peptide across the samples. That's precious. And I really like being able to couple our like new and old or recently synthesized or old protein with the power of the quantitation in the MS3 rather than relying on reconstructed chromatograms.
Thanks. How about Alexi or Tomas?
For us, there are 2 main benefits. First one is the quantitation precision. You can never match it with any other method. And you get it for free for 16 or 18 samples. And another thing is the depth of the quantified proteome. By using the multiplexing and the mass tag, you can fractionate your sample indefinitely. You can spend infinite time in analyzing your 16-plex or 18-plex and go as deep into the proteome as possible, maintaining the quantitation precision, the one you wanted. And until recently, the throughput was also the thing. You could analyze in 24 hours 16 and 80 samples to the pretty good depth, which is now challenged by the label-free and data-independent acquisition approaches.
All right. How about we can go to, we got another question here. And maybe Steve, you can talk about this since you had mentioned this in your talk as well. In the past decade, there's been some major advances, I would say, in LC-MS instrumentation. And specifically, we now have the Orbitrap Astral system, as well as these advanced tribrids like the Ascend. I mean, can you comment on how these have improved proteomic analysis and multiplexing?
I mean, I don't really know if there's a way to comment on how amazing they really are. It's remarkable to think. I think the jury's still out, but I think a year from now, the impact of the Orbitrap Astral will finally kind of rear its head, and we'll know exactly how amazing it really is. For right now, we think it's probably at least an order of magnitude better than what we were seeing in the past. So I feel like it's gigantic, right, in its ability. So we went from working as hard as we could and having about 9,300 proteins that we could quantify to having about 11,400 proteins quantified. So an additional 2,000 proteins that we had never seen before in these analyses. And they just came right out with the Orbitrap Astral. So yeah, I think a lot deeper data sets, a lot more sensitivity. So yeah, it's really hard to say what the impact will be, but I think it'll be huge.
All right. Yeah, no, thanks on that. And maybe Dustin, I know, you're in my group and you do a lot of reagent development. I mean, what do you think about the future of these formats as well as higher plexing? Maybe you could just comment on that.
Yeah, so now that I'm involved with the development of TMT technology and future TMT products, so I'm particularly excited for the potential that exists to increase multiplexing. It ties in with the previous question with just simply high resolving power on Orbitrap mass analyzers, which enabled 6-plex TMT to go to 11. It enabled 18-plex TMT Pro. We know that's not the limit. So we've hinted at previous conferences this year at, there's higher potential for that. We haven't reached it. We might be able to go beyond 30-plex just with different isotopic variants. Not to say too much more about that, but that's what's got my attention lately.
Thanks. All right. So I'm looking to see if there's some more questions came in. So I guess we have time for one more question. And again, I'll go around to each speaker so they get a chance to comment. But I guess we heard Dustin talk about, he's excited about the future of higher plexing. But is there anything else that you guys have in mind in terms of TMT technology that gets you really excited? We think we've seen this technology applied in multiple different ways. So maybe we'll just go around again to everybody. And I guess I'll start off with, Fernando. I don't know if his audio is working. I haven't heard from him yet, but we could start with him, or we could go to, his colleague, let's see, Alberto, maybe you can comment about what excites you about the future of using mass tags.
Well, I'm coincident with Dustin, in which I think that the future of the labeling technology passes by increasing the number of plexing. Because now DIA is hitting hard. I mean, it has supposed a big advance in the field, and the way that TMT should fight against it is increasing the number of plexing, in my opinion. So some advances that have been exposed here are very interesting in this aspect.
Thank you. I agree. I think that's going to be really exciting if as those tags are developed. Let's see, Alexi, do you want to make a comment about the future of plexing in your work?
Yeah, I think increasing of the number of channels is the obvious one, and we all want to have a 96 channel to go like full plate on. But another thing which I think will play a bigger role is the intelligent acquisition, because the main benefit of TMT right now is that within the 16-plex or 18-plex or future 30-plex, you have virtually zero missing values. If you can tweak your data acquisition with increased speed of astral detector whatsoever, so you always sample the very same peptide population from the multiplex to multiplex, then you can project this virtually zero missing values to a number of multiplexes. And then you hit tremendous proteome coverage, zero missing values, and extremely good quantitation precision. So that's what I'm looking for.
No, thanks for that. I think whether you use label-free or labeled samples, the batch effect is real, and the matter of basically being able to predict and also accommodate for those differences and have fewer missing values, I think will really drive for a more complete measurement of the proteome. So we'll also go to Jeff Savas, you got any comments about the future of multiplexing? I know you kind of have this hybrid SILAC or metabolic labeling with TMT, but what do you think the next step is for your research?
I think I'm excited about micro scaling the amount of reagent needed in the smaller quantities of samples. So for a whole proteome, we can get 50 micrograms, no problem, but some samples we can't recover that amount. So it'd be exciting, something with solid phase or some type of a high efficiency labeling reaction that we could run at a smaller scale. And I'm also very interested about combining TMT with other technologies like cross-linking or other types of chemical probing to actually couple multiple tools into the same analysis run.
Oh yeah, thanks. That's actually a great point. I think PTM analysis, structural biology, those are definitely great applications of using mass tags to try to increase throughput to look at protein dynamics as well as protein modification. And again, maybe we'll end here on Steve. Maybe you have some comments about the future. I think you presented some really exciting things about, again, higher plexing and targeted proteomics, but just your thoughts as we close out this session.
Yeah, thanks, Ryan. I'm really excited about the opportunity for targeted proteomics. I feel like it's pretty enormous because you can have thousands and thousands of samples, and if you can prepare them on an acceleromes or some other way to do sample prep, then you've got this opportunity to really look through really high numbers of samples. And being able to do that in a targeted way would be, very exciting. I also think about what Alexi said, that maybe some hybrid strategies may actually come about for TMT, where you have a proteomic experiment, but also you want to catch the bottom 10% in every single plex. So there could clearly and probably easily be ways to combine some aspects of targeted and untargeted proteomics in order to guarantee that you get much higher numbers of samples and much better coverage.
All right. Well, thanks, Steve. Well, I think we're at time for our session. I wanted to thank all the speakers for some great presentations today. I also want to thank the audience for participating and with the Q&A. Again, just a reminder that these webinars that were presented will be available on demand after today. And again, I just want to thank everybody again for joining a great session.
Thanks.
Bye.
Thanks for having me.